We all know that photosynthesis happens during daytime, and that is because a part of the process is light dependent, meaning they need sunlight in order to create reactions that are needed to execute photosynthesis. However, in cellular respiration, it happens mostly nighttime, and in some cases happens during daytime. Cellular respiration is the process by which organisms combine oxygen with foodstuff molecules, diverting the chemical energy in these substances into life-sustaining activities and discarding as waste products carbon dioxide and water. Hence, cellular respiration in plants is the process to convert the glucose made during photosynthesis into energy in the form of adenosine triphosphate, or commonly known as ATP, which fuels the plant's cellular activities. Now let's dive deeper in the world of cell respiration.
The three processes of cell respiration are the following. First, glycolysis. Second, Krebs Cycle. Third, oxidative phosphorylation.
So for glycolysis, as the first process takes place in the cytosol of a cell, and it can be broken down into two main phases, which are the energy-requiring phase and the energy-releasing phase.
In energy-requiring phase, molecules of glucose start to get rearranged, and two phosphate groups are attached to it. The phosphate groups are made to modified sugars, now called fructose 1,6-biphosphate unstable, allowing it to split in half and form two phosphate-bearing 3-carbon sugars. Because the phosphate used in these steps come from ATP, two ATP molecules get used up. The 3-carbon sugars formed when the unstable sugar breaks down are different from each other. Only one glyceridide and 3-phosphate can enter the following step. However, the unfavorable sugar DHAP can easily converted into the favorable one. In the energy-releasing phase, each 3-carbon sugars is converted into another 3-carbon molecules pyruvate through a series of reactions. In these reactions, two ATP molecules and one NADH molecule are made. Because in this phase takes place twice, once of each two 3-carbon sugars, it makes 4 ATP and 2 NADH overall.
Each reaction in glycolysis is catalyzed by its own enzyme. The most important enzyme for regulation of glycolysis is phosphofructokinase, which catalyzes formation of the unstable 2-phosphate sugar molecule, fructose 1,6-biphosphate. Phosphofructokinase speeds up or slows down glycolysis in response to the energy needs of the cell. Overall, glycolysis converts one 6-carbon molecule of glucose into two 3-carbon molecules of pyruvate. The net products of this process are 2 molecules of ATP and 2 molecules of NADH.
Now we have the Krebs cycle. The two 3-carbon molecules of pyruvate will enter the matrix of mitochondria and start the Krebs cycle.
But wait, before the Krebs cycle begins,
Pyruvic acid, which is just the 3-carbon molecules of pyruvate, splits apart and combines with an enzyme known as CoA, which stands for coenzyme A. The product of this reaction is a 2-carbon molecule called acetyl-CoA. The third carbon from pyruvic acid combines with oxygen to form carbon dioxide, which is released as a waste product. High-energy electrons are also released and captured in NADH. The Krebs cycle itself actually begins when acetyl-CoA combines with a 4-carbon molecule called OAA, also known as oxaloacetate. This produces citric acid, which has 6 carbon atoms. This is why the Krebs cycle is also called the citric acid cycle.
The final step of the Krebs cycle regenerates OAA, the molecules that began from the Krebs cycle. This molecule is needed from the next turn through the cycle. 2 turns are needed because glycolysis produces 2 pyruvic acid molecules when it splits glucose. After the second turn through the Krebs cycle, the original glucose molecules has been broken down completely. All 6 of its carbon atoms has been combined with oxygen to form carbon dioxide. The energy from its chemical bonds has been stored in a total of 16 energy carrier molecules. These molecules are 4 ATP, includes 2 from glycolysis, 10 NADH, includes 2 from glycolysis, 2 FADH2.
Lastly, we have the oxidative phosphorylation, which is basically the need of oxygen. It is made up of 2 closely connected components, the electron transport chain and chemiosmosis. In the electron transport chain, electrons are passed from one molecule to another, and energy released in these electron transfers is used to form an electrochemical gradient. In chemiosmosis, the energy stored in the gradient is used to make ATP.
So where does oxygen fit into this picture?
Oxygen sits at the end of the electron transport chain, where it accepts electrons and picks up protons to form water. If oxygen isn't there to accept electrons, the electron transport chain will stop running and ATP will no longer be produced by chemosis. Without enough ATP, cells can't carry out the reactions they need to function, and after a long enough period of time, may even die.
The electron transport chain is a series of proteins and organic molecules found in the inner membrane of mitochondria. Electrons are passed from one member of the transport chain to another in a series of redox reactions. Energy released in these reactions is captured as a proton gradient, which is then used to make ATP in a process called chemiosmosis. Together, the electron transport chain and chemiosmosis make up oxidative phosphorylation. Delivery of electrons by NADH and FADH reduce electron carriers from other steps of cellular respiration, transfer their electrons to molecules near the beginning of the transport chain. In the process, they turn back into NAD+ and FAD, which can be reused in other steps of cellular respiration. Electron transfer and proton pumping. As electrons are passed down the chain, they move from a higher to a lower energy level, releasing energy. Some of the energy is used to pump H+ ions, moving them out of the matrix and into intermembrane space. This pumping establishes an electrochemical gradient, splitting of oxygen to form water. At the end of the electron transport chain, electrons are transferred to molecular oxygen, which splits in half and takes up H+ to form water. Gradient-driven synthesis of ATP. As H+ ions flow down their gradient and back into the matrix, they pass through an enzyme called ATP Synthase, which harnesses the flow of protons to synthesize ATP. That's all for now, and we remind you to always keep learning. Bye!